Happy Monday, everybody, and welcome back to another wonderful day of online living environment. So today I'm going to be showing you guys how to do the virtual lab that we've posted on Google Classroom. So you're just going to need a few things to complete this lab. You're going to need paper of some sort. Now this can be just plain white paper, this can be loose leaf, this can be random scrap paper you found lying around your house, up to you. Whatever you can find in your apartment that you can write on. You're also going to need colored pencils. Now, if you don't have colored pencils, you can also use markers or colored pens, anything that is going to give your drawing some color. You're also going to need scissors. Don't run with these. And you're also going to need either tape or glue. Now, I don't have glue at my apartment, so I'm going to use tape. So that's all you need for today's lab. You're also going to need the paper that I have posted on Google Classroom. It's called DNA Lab PDF. Now the very first thing you're going to do before you even complete this lab is you're going to read the passage at the top. Where is this? You're going to read through this passage at the top of the PDF. Okay. This passage is going to overview everything you need to know about DNA. It talks about the structure of a nucleotide. It talks about everything you need to know for DNA, essentially, for your test that is when? On Wednesday. So make sure you are reading and annotating this passage. You are going to turn this in, so I want to see that you have annotated and thoroughly read that passage. Now, as soon as you are done reading the passage, you're going to see a page that looks like this. This is where you'll find your materials and your procedure. Now, we just went through the materials, but I'm going to go through this procedure with you guys. So the very first thing you're going to do is, if you have a printer at home, you're going to print out this page. These are the different structures that you're going to use to assemble your DNA. However, if you don't have a printer, like I don't, you're going to be drawing those different components on a sheet of paper. Okay. So if you do have a printer, feel free to skip forward to the next part of this video. But if you don't have a printer, stick around with me and we're going to draw some, okay? So the very first thing you're going to do, you've got two options of how you can do this. You can either trace these different components by using your phone as a template, or you can just freehand it. So if you are really artistically challenged, maybe trace this instead. So what you're going to do is, once again, access that PDF on your phone. You're going to zoom in to one of these components. So right now I'm looking at my deoxyribose. You're going to hold that. Oh, my PDF moved. Hold on. Wait for it. There it is. All right. You're going to hold that that picture up to the piece of paper. Where is it? And then you can color straight on this paper and trace it on your phone. But I'm not going to do that. I'm just going to freehand it. So we'll see how this goes. We all know how artistic I am. So the very first thing I'm going to draw is my deoxyribose. Okay. I'm going to show you how to draw one of each of these, and then we'll fast forward through the rest as you draw all, I don't know how many components there are, 15. That's just a ballpark number. So my deoxyribose needs to be purple. So I've got my purple colored pencil, and I'm going to draw the shape that I see on my PDF. Okay, it looks something like this. No, that is not great, but it is as good as it's going to get. Alright, so you can see that my deoxyribose is going to look like this. Okay. Now I'm going to actually have to draw 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 deoxyriboses. So I'm not going to show you how to draw all 12, but this is what your deoxyribose shape is going to look like. Okay. The next thing I'm going to draw is my guanine. Now my guanine is going to be blue. So I've got my blue colored pencil, and I'm going to copy down my guanine shape that looks like that looks like this. Okay, so there's my guanine. Okay, the next thing I'm going to draw is my adenine. Now my adenine is going to be red, so I'm going to draw that. Alright, and there is my adenine in all its glory. Alright, the next thing I'm going to draw is my cytosine. So my cytosine is yellow. Alright, my cytosine looks like that. Next up, what am I missing? Let's see. Oh, my thymine. I've got to draw my thymine. Now, thymine is green, so I've got my green colored pencil. And I'm going to draw thymine looking like... Oh no, I messed up. Alright, and my thymine looks like that. I messed up, but I'm going to cut that off. And now, finally, the last thing I need to draw is my phosphate group, which is the last component of our nucleotide. So my phosphate group is brown, and it looks like a plus sign. So I'll draw it right here. Oh, that is atrocious. Please be neat as you're going through this. It'll help you in the long run. Alright, and here is my phosphate group. So these are all of the general shapes that you are going to need to copy onto your paper. Now, I actually have to do this, and you do too, several more times. So I need to have 3 guanines, 3 cytosines, so basically 3 of each base, and then 12 phosphate groups, and 12 deoxyriboses. So I'm not going to do that, but I'm going to fast forward this video so you don't have to sit here and watch me color each one of them. However, right now I want you to pause this video and draw each of these shapes on your paper. So once again, this is not a free-for-all. You are copying down the shapes that you see in this PDF file. You're going to copy them exactly how you see them, and please label them with their appropriate name. Okay, so now I'm going to draw the rest of these. Here we go. Alright, so I just finished drawing all of my components. This is pretty beautiful, if I do say so myself. I did mess up quite a few times, but that's okay. So now what I'm going to do, and you're going to do as well, I've really struggled with that thymine, is you're going to color these in. So we've just drawn the base, or we've drawn the backbone of them, but you need to color them in entirely in that color that was assigned. So let's see if I can show you what that's going to look like. Alright. So you're going to shade your bases in entirely. This is going to be the same if you printed them out or if you've colored them like I have. You're just going to take your colored pencil and lightly shade in each one of these bases. Alright, so there's our beautiful deoxyribose. There's our beautiful guanine. There's our beautiful adenine. There's our cytosine. There's our thymine. And there's our phosphate. Alright, so once again, you are going to do this for every single one of those bases. So I'm going to click fast forward again as I color the rest of these, and you can do the same. Alright, so I just finished coloring everything. Looks fantastic. I'm an artiste, if you will. So now the next step that I and you are going to do is we are going to cut these out. So you should have grabbed some scissors at the beginning of this tutorial. Now is when we're going to use them. Please be careful as you cut these out. I don't want you to have to start over. But you are just going to, let's see if I can show you how to do this. You are going to neatly cut out along the line that you have drawn. Now once again, this is the same for if you printed out this copy. You're just going to cut out along the lines that you printed off. Okay. This is going to get messy, so please make sure you clean up your papers after yourself. Don't make mom or dad clean it up for you. Alright, so we're going to cut these out. And you're going to be left with several of these beautiful little shapes. Please keep these off to the side. We are going to use them in a minute. Don't lose any, okay? Now once again, I am going to speed up this video as I cut out the rest of them, but it is up to you to cut out each of your shapes that you'll use to assemble your DNA. So once again, let's speed this up. We're going to cut some more. I gotta take a break. Alright, let's get back to it. And I am officially done. Now, I don't know about you, but my hand is cramping. Now that I have cut out all of my pieces, you should have 12 deoxyribose, 12 phosphate groups, 3 thymine, 3 adenine, 3 guanine, and 3 cytosine. Okay, so I've got all of my pieces separated by color over here. Now, I'm going to take a break and clean up because I've got papers everywhere, and you should probably do the same. So once again, I'm going to fast forward as I clean. Alright, I'm feeling much better about that. Okay, so now if you look at the procedure section of your paper, the next thing we're going to do is we're actually going to build a nucleotide. Oh, I'm out of breath. Oh my god. So this is going to be our model nucleotide. Now, if you remember from our lessons, nucleotides have 3 components. The first component is a deoxyribose, right? We need that sugar, okay? So I'm going to tape or glue that deoxyribose onto a sheet of paper. Okay, this is going to be my template. I'm going to have my nucleotide at the top and my my DNA at the bottom. Okay, so you're going to take 1 deoxyribose, and that is going to be our first part of the nucleotide. Okay, where's my paper? Okay, I'm going to glue that down right here. Okay, now the next part of our nucleotide is going to be a phosphate group. So I'm going to glue that. I'm actually going to move this down because I need to put my phosphate group at the top. Okay, so there's my deoxyribose. All right, then my phosphate group is going to serve as a bond between different nucleotides. Okay, so I'm going to put that phosphate group right on top of that deoxyribose, like this. And then if you remember correctly, the very last thing you need for your nucleotide is a base. Okay, so I want you to just pick your favorite base, unless does it specify? I don't think so. Nope, so just pick your favorite base, maybe one that you have an extra of, and glue it, attach it directly to your deoxyribose. Okay, so it's going to look something like, let's see, let's do it right there. I'm going to attach my adenine. Yeah, let's do adenine. Might as well. Okay, so I'm going to take my adenine right here, and I'm going to tape it to my deoxyribose like this, or glue it, either works. Okay, so your nucleotide, this is really ugly. That's better. Alright, your nucleotide is going to look like this. So you have the three components of a nucleotide. You have a phosphate group, a deoxyribose sugar, and you have your base. Okay, we're now going to use that as an example to construct our full DNA strand. So once again, you have an example of what this should look like on your PDF. We're going to make that using our nucleotide as an example. Okay, so I'm going to label this on my paper and say that this is our nucleotide. Okay, so here is our nucleotide. Now we're going to create an actual DNA strand. So the first thing I think it tells us to do is to construct the right side. Yes, construct the right side of our DNA. Okay, so it wants us to put in sequence a cytosine, thymine, guanine, and adenine. So we're going to do this 4 times with a C, a T, a G, and an A on the right side of our molecule. So let's do it. Okay, the first thing you need to do is, once again, we're going to use a phosphate group. Okay, we're going to have that at the top side of our strand. I'm actually going to do this on a new sheet of paper. Okay, and we're going to attach a phosphate to a deoxyribose. Now, something that you might notice is that you actually have to put or connect this deoxyribose upside down or backwards. We're going to get to why that is in just a minute, but let me glue these together. I'm struggling. Okay. So the right side of your strand is going to look something like, let's see, arts and crafts is not my strongest suit. Okay, and I'm going to do a cytosine first. There it is. Beautiful. Your first nucleotide is going to look something like this. You'll notice that my deoxyribose and my phosphate are upside down from what it should be. We'll get to why that is in just a minute. Okay, so there's my first nucleotide. Now the next one it told me to add is a thymine. So I'm going to do that same thing. I'm going to attach a deoxyribose. I wish you guys could see what I'm doing. Oh, there it is. Okay, I'm going to attach a deoxyribose. I'm going to attach a phosphate group. And then I think it told me to add a thymine. Is that correct? Yep, thymine. Okay, so then I'm going to add my thymine. Okay, there's my second one. Now I'm going to do that again, and this time we're going to add a guanine. So once again, I'm going to take my deoxyribose and glue it down right underneath my phosphate. I'm going to add another phosphate group. And then I'm going to add a guanine. So I'm going to grab a G. Alright, and now finally our last base for the right hand side is going to be an adenine. Okay, so we're going to do the same thing. We're going to add a deoxyribose to make our backbone. We're going to add a phosphate to link it together. And then we're going to add a, what are we missing? An adenine, is that correct? Yep, and an adenine. Okay, and we're going to glue that just like that. Alright, so now I have finished the right-hand side of my DNA. Yours should look something like this. I hope it looks better than mine, because mine is kind of tragic. But you are going to glue together your deoxyribose with phosphate bonds, connect that backbone together, and then your bases are present connected to the deoxyribose. Okay, so this is the right side of your strand. Now we're going to use our base pairing rules to make the opposite side of the strand, because remember DNA is double-stranded, which means we're going to have 2 copies of this, one on either side. Okay, so now our next part of our procedure tells us to complete the left side of the DNA ladder by adding complementary nucleotides or nucleotides that fit. So our very first base that we have at the top is C. So what do we pair with C? I hope you said G. You also can figure it out because our bases are going to connect with the same shape as the one that we have already connected. Okay, so I'm going to connect a G to our C up here at the top. Now, there's our base, right? There's our base, but then what do we need to connect to that base to continue on our backbone? Well, I hope you picked out one of your deoxyribose, because you're right. We're going to connect that deoxyribose to our base, to our G. Now, at the very bottom of our deoxyribose, we need to add a phosphate group. So take out one of your lovely P plus signs, and you're going to connect it so it looks like this. Okay, so now we're making the second side of our DNA strand. My shapes were a little bit off because I can't draw, but we connected these 2 bases based on shape. Our G and our C connected. We then we added our deoxyribose group as well as our phosphate bond. So you might notice that this time our deoxyribose is facing upright. It's not upside down this time. That is one of your questions, so I want you guys to figure out why that's the case. Alright, let's keep going. So we're now going to add another deoxyribose group to our phosphate bond, and we're going to connect the corresponding A, or adenine base, to our thymine. Okay, so I'm going to connect my A first to my T, and then I'm going to bring in a deoxyribose and connect these 2 together. Now, my drawing was not too great, and my deoxyribose is kind of overlapping my A, but we're going to press on regardless. Let's see if I can fix this. Okay. All right, let's see if that's a little bit better. Ooh, not quite, but it's gonna have to do. All right, that's better, I guess. All right, so then I've connected my second nucleotide, okay? So I connected my A to my T. I added my deoxyribose and my phosphate bond to link the two. Okay, now I'm going to do it again. I'm going to add a cytosine base and connect it to my guanine. So once again, we see that those shapes connect, so it's really easy to match them up. Okay, so I've connected my bases. Now I'm going to connect my deoxyribose to my base. I'll show you guys what that looks like. So I just connected my bases together. Now I'm going to add my deoxyribose. Oh no, my spacing is really off. All right, and then finally, you're going to add a phosphate group to connect the two. All right, so this is what, let me add there. So now this is what mine's looking like so far. It's not very neat. So as you do this, please be neater than I am. If you printed yours off, it should be pristine. If you are drawing yours like me, we'll cut you some slack because I know how hard this is. But try to make this as neat as possible. If your shapes don't match up completely and you drew yours like mine, we'll cut you some slack, okay? But I have one more to go. So now my very last base that I need to connect, we have an adenine, so I'm going to match up a thymine. So once again, we see that those shapes match up completely. So I'm going to tape ours together like that. Okay, now I'm going to add one last deoxyribose. Let's make it pretty. So adding one last deoxyribose, and I'm adding one last phosphate group. Oh no, my phosphate's too small. Alright, I'm connecting my very last phosphate, and there you have it. I'm going to connect one last phosphate group at the, actually, nah, I'm not going to. Alright, so this is what your finished DNA strand should look like, okay? Notice that we have a deoxyribose on both ends, we have phosphate groups to hold them together, and then that pattern's going to repeat. So this is what makes up our backbone of DNA. Remember we described it kind of like a ladder shape because these bases in the middle are what make up the rungs of the ladder. We see that A matches up with T, C matches up with G, and etc. So this is your completed DNA strand. I'm going to label mine at the bottom. So this is my DNA strand. Okay, so this is my double-stranded DNA. Here's just my single nucleotide. This is what your finished product should look like. So now you're going to use these models to answer the questions that are on your PDF. So if you scroll to, let's see, the bottom page, there are a few questions that you are going to answer. There's not enough space to write these on the PDF, so please write these on a separate sheet of paper. And once again, you're writing them in complete sentences, okay? Now, when you submit this work onto Google Classroom, I want you to take a picture of what your finished product looks like. So you can use your phone. I hope you know how to take a picture. You're going to use your phone, take a picture of that DNA strand, and then you will upload this picture. Upload this picture to Google Classroom. Okay, upload a picture of both your DNA strand and your nucleotides, as well as your completed questions. All right, if you have any questions, please let me know, but that is how you complete your wonderful DNA model. Now, when you're done with this, feel free to frame it in your room. I will definitely be hanging mine up so my roommates can enjoy it because this is a work of art. All right. Yeah, if you have any questions, let me know. Other than that, have a wonderful day, guys. This is due tonight at midnight, along with your Genetics 2 homework that was assigned this weekend. All right. Have a good day, and I'll see you guys tomorrow.